in Martin Simon's lab in Quebec City. And this is one of the projects I've been working on in the last years. So today I'm going to talk to you about how we found a new role for the RabGAP TBC-11 in microRNA function. So in the lab, we're quite interested in microRNAs. So let me just explain first where these small molecules come from. So microRNAs are first transcribed in the nucleus by RNA polymerase II, and they are transcribed as these long transcript called primary microRNAs. And then they will undergo a first processing step in the nucleus by the enzyme called DROCHA, and this will generate a precursor microRNA, which can then be exported to the cytoplasm, where there will be a second processing by the enzyme called dicer, and this will generate a microRNA duplex. Then this duplex, it can be loaded onto a protein called the Argonaute, and only one strand will be kept in the Argonaute, and this will form what we call the minimal mRISC, and the other strand, which we call the passenger strand, will just be degraded. So in the lab, we've been focusing mostly on this effector complex that we call the mRISC. So mRISC stands for microRNA-induced silencing complex, and it is composed of a microRNA that you can see in red in the picture. And this microRNA is bound by an Argonaute protein, which in C. elegans, we have ALG-1 and ALG-2, who are the Argonaute proteins who are specific to the microRNA pathway. And then sometimes you have a GW182 protein, which in C. elegans are called AIN1 and AIN2. These proteins are some scaffold proteins that are recruited directly by the Argonaute. So through partial base pairing with the target mRNA, the microRNA can therefore recruit the mRISC on a target mRNA. And then the mRISC can induce many mechanisms to repress this target mRNA. So you can have deadenylation of the target mRNA, which is mediated through the CCR4 and the PAN complex that are recruited by GW182. You can also have some decay of the target mRNA. So this can either be from the 3' end after the deadenylation, or from the 5' end after decapping, which is mediated by the DCP complex. And finally, the mRISC can also induce translational repression. However, the exact mechanism on how this is achieved is still under debate. So we know that the RISC can do many things. It can induce different mechanisms, but one question we've been asking in the lab for a while now is how can we modulate the activity of this complex? And so what do we do in C. elegans when we have such a broad question? We do a genetic screen. So this is what we did. We performed a forward genetic screen in order to identify new regulators of the mRISC activity. And so briefly, we developed this reporter system where we have GFP, in which we inserted these box B sequences in the 3' UTR. And we also have a ALG-1, so the Argonaute protein, which is fused to the lambda N peptide. And this, the lambda N box B, will recruit artificially the Argonaute on the 3' UTR of the GFP. And this is sufficient to repress the reporter. So what we did is we did a screen to identify mutants in which this GFP repression was affected. So there was derepression of the GFP. So you can see here on the left, this is the silenced GFP in the initial strain. And then on the right is one of our candidates that we obtained from the screen in which we saw a GFP derepression. And so we mapped this mutation to be in a gene called TBC-11. And the mutation we isolated, we named the allele QBC24. So TBC-11, as it says in my title, is a RabGAP. So it's a RAB GTPase activating protein. So it acts on the RAB family protein, which are proteins that are implicated in vesicular transport in the cells. And so these RAB proteins, they alternate between a GTP active state and a GDP bound inactive state. And this switch between active and inactive is catalyzed by enzymes that are the GIF and the GAP. And so TBC-11 is a GAP. Therefore, it acts on RAB proteins to inactivate them. And so why did we think this was interesting that we found this type of protein in our screen is that there has been in the last years a number of studies that shown that microRNA function is linked to vesicular trafficking and to the endomembrane system. And so many groups have shown that the MVBs, the ER, and the Golgi are implicated in proper microRNA activity. More precisely, what these studies have shown is that the mRISC components and microRNAs, they associate with the multivesicular bodies, the MVB. The microRNA loading and target repression can occur at the rough ER. And lastly, a study that was actually done in our lab a couple of years ago showed that the Golgi-associated retrograde protein complex called the GARP, and this plays an important role in microRNA function. And so we'll come back to this paper later. So knowing this, that there was a link between microRNA activity and the vesicular trafficking, and knowing that we had identified a RabGAP in our screen, we set out to answer these two main objectives. So the first one was to confirm the implication of TBC-11 in microRNA function. We did this by looking at microRNA-related phenotypes and also through microRNA activity reporters. And our second objective was to characterize the molecular effect of TBC-11 on the mRISC. And so to do this, we started by identifying the implicated RAB protein. We looked at the mRISC association to endomembranes. And finally, we studied the intracellular localization of ALG-1. So for the first objective, I should start by saying that throughout this project, we've been working with two alleles. So this is the TBC-11 protein. So the allele that we isolated, the QBC24, is a SNP in the catalytic domain of the protein. But there was also on CGC a deletion allele that's presumed to be a null. So we ordered this one and we characterized both in parallel. So the first thing we looked at was the seam cell division pattern. And so seam cells are these hypodermal cells on the animal, which undergo asymmetrical divisions throughout the life of the animal. And at the end of the L4 stage, you are left with 16 seam cells, which will then, on the transition to adulthood, they will fuse to form this structure that we call the allele. And why this is interesting to us is that these divisions are tightly regulated by the LIT7 microRNA family. So for us, this is a readout for microRNA function. So the first thing we did was to count the number of seam cells in adult animals. So you can see wild-type animals have an invariable number of 16 seam cells. However, in both our TBC-11 allele, we could see a range from 14 to 18 seam cells, so indicating that there was defects in this division program. And then we scored, we looked at the allele structure. So when you have an incorrect number of seam cell, the fusion will not happen properly, and you will have these breaks in the allele. So this is what we scored here. So we could see that in both TBC-11 alleles, we do see breaks in the allele. So indicating that TBC-11 affects the normal seam cell division pattern. Then we looked at another phenotype that's associated to LIT7. So we wanted to see if we had a genetic interaction with the LIT7 microRNA. So LIT7 mutants, they are 100% lethal, but we do have a sensitized background, which is a TS allele, so it's 100% lethal at 25, but it can be maintained at 15. And the animals, they burst at the L4 to adult transition, and this is caused by a defect in LIT7. So this are animals that were kept at 15 degrees. And you can see this LET7 sensitized background has about 35% of bursting animals. However, when we do a double mutant with TBC-11, this percentage is increased. So indicating that TBC-11 can enhance the LET7 family phenotypes, so both the bursting and the allele. Then last thing we wanted to check for this objective, so we wanted to see if this effect was specific to the LET7 family of microRNA, or if it was affecting other microRNAs. So we looked at this miR228 activity reporter. So the reporter is just a GFP that's fused to a histone. So the signal is accumulating in the nucleus, and it's under a promoter that will express it in the intestine cells. And this reporter is repressed by miR228. So we checked again the effect of both allele of TBC-11 on this reporter, and you can see that there is de-repression, which is quantified here on the right. And so indicating that TBC-11 is not just affecting the LET7 microRNA family, but it's also required for proper repression by miR228. So now that we've confirmed that TBC-11 had an effect overall on microRNA function, we wanted to characterize what was the molecular effect on the mRISC and why was it causing these microRNA defects. So first thing we wanted to test was, well, to identify on which RAB TBC-11 was acting. So different gaps can affect different RAB proteins, which are all implicated in different steps of vesicular trafficking. And so we wanted to identify the RAB that was targeted by TBC-11 in this case. But since there was very few information known about TBC-11 in C. elegans, we turned to its human ortholog, which is TBC-1D11. And it is known in humans that TBC-1D11 is a gap for RAB2 for 6, 11, and 36. So these were good candidates to test. And we started by testing RAB-6 because, as I mentioned, all RAB proteins are associated to different steps in vesicular trafficking. And RAB-6 is known to be implicated at the Golgi. And as I mentioned earlier, we had previously shown in the lab that there was a link between the transport from the Golgi and the microRNA-mediated gene regulation. So we started by testing if TBC-11 was a gap or was acting on RAB-6. And to do this, we based our experiment on the principle that in absence of TBC-11, there's no more gaps, so the RAB protein is maintained in a hyperactive state. And this is what causes your phenotypes. So if you could decrease the level of the RAB protein with RNAi, for instance, then you should be able to suppress your observed phenotype. So this is what we did and what we saw. So we were scoring here allele defects as a readout. And as you can see in TBC-11 animals, you have these defects that are suppressed when the animals are exposed to RAB6.1 RNAi. And this was also the case with RAB6.2 RNAi. So from this, we could conclude that TBC-11 was acting through RAB6 to control microRNA function. So next thing we wanted to assess was how is it affecting the mRISC. So the first thing we did was to do just Western blots on the level, to see the level of the mRISC components. And this is when we started noticing that something was happening, because when we were just doing experiments where we would just quickly boil some worm in the SDS and load that on the gel, we wouldn't see major striking differences, at least in ALG-1, in the levels in our TBC-11 alleles. However, when we prepared worm extract, so when we had worms in worm lysis and then centrifuge and load that, then we would see differences. And so in one of our allele, we saw less ALG-1, whereas in the other allele, we saw more. So we wondered if this could be associated to the fact that when you do worm extract with detergent, you are limited in your ability to extract membrane-associated proteins. So what we did is we repeated the experiment, but this time doing worm extracts with some lysis buffer that had different concentration of detergent. So we did with 0.5% detergent, which is our normal extraction condition. And we also did the experiment with 1.5% detergent, which should allow you to extract more membrane-associated protein. So first, if you look at the 0.5, we do again see that we have less ALG1 in the QBC allele and more in the OK. And here I'm just showing LG1 because N1 did not follow this trend. However, when we used more detergent, so we were extracting more membrane protein, we could recover more ALG1 in our QBC allele, whereas we had less in our OK allele. And so we could conclude that both alleles did affect the association to endomembranes. However, they have opposite effects. So the OK allele has less LG1 associated to endomembrane, whereas the QBC allele has more. And we hypothesized that these two alleles behave differently because the OK allele is presumed to be a null, so there's no protein expressed, whereas our QBC allele is just a SNP in the catalytic domain. So the protein is still expressed, and we hypothesize that it can still bind the RAB protein, but it is inactive, so it kind of traps the RAB and its cargo protein on the endomembrane. So to test this hypothesis, if there was really more LG1 trapped on endomembranes in our QBC24 animals, we wondered if we could see it if we were looking at a CRISPR tag GFP LG1. And so we looked at the localization of LG1 in the seam cell of these animals. So what you can see here in wild type, you can see the seam cells, which are these nice elongated cells throughout the animal, and you can see that LG1 is expressed throughout the cytoplasm of these animals, of these seam cells. However, when we looked in our QBC24 mutant, what we saw is that we did not have a nice signal of LG1 throughout the cytoplasm, whether we had a accumulation of, or the LG1 signal was concentrated around the nucleus. And so we wondered if we could rescue this with RAB-6.1 RNAi, and we could. So you can see that when the QBC24 animals are exposed to RAB-6.1 RNAi, you once again see this expression of LG1 throughout the cytoplasm. So we wanted to identify what was the structure we were seeing. So we tried co-localization with a Golgi marker. However, it did not co-localize with that. And then we tried several times to express the rough ER marker TRAM1 in these cells. However, after many attempts, we were never able to get expression of this marker in the seam cells. So we could not directly obtain co-localization images. But what we did do, however, since we were we had the hypothesis that this was the rough ER, we monitored the association of LG1 to polysomes because there is a high density of ribosomes at the rough ER. So we wondered if we could see a difference in LG1 association to ribosomes. And so to do this, we did polysome profiling in wild type and TBC-11 animals. And then we blotted LG1 in the different fractions. And what you can see here is that in the TBC-11 animals, you do see LG1 in these fractions that correspond to the heavy polysomes, whereas we don't see as much LG1 in wild type in these fractions. And this is quantified here on the right. So from this, we could conclude that LG1 is more associated to heavy polysomes in our TBC-11 animals. And so the last thing we wanted to answer, so we were wondering if there is more LG1 that's accumulating on these polysomes, why is it not effective? Why is it not repressing targets because we could see early on that we had defect in microRNA-mediated silencing. So we wondered if this ALG-1 that was accumulating was associated or not to microRNAs. So to do this, we did immunoprecipitations of ALG-1, once again using low and high detergent concentration. And then we quantified the associated microRNAs by qRT-PCR. So what you can see on the left in wild type, in blue is the level of ALG-1, that the relative level that we could IP in high detergent compared to low detergent. And there was no difference in the quantity of ALG-1 we could IP, and there was no difference either in the level of associated microRNAs. However, in the QBC24 animals, when we extract with high detergent concentration, we could IP more than twice more ALG-1 in these conditions, as you can see here on the plot. But however, the level of microRNA associated to ALG-1 was not higher in these concentrations. So indicating that it's an unloaded form of ALG-1 that's accumulating to endomembranes in the TBC-11 QBC24 animals, and that is why they can't properly repress their targets. So we'd like to propose this model where in a normal cell, ALG-1 needs to be transported in different locations within the cell, and that this transport could be mediated by different RAB proteins. However, RAB-6 is known to mediate the transport between the Golgi and ER. So in normal conditions, it would be responsible for, with TBC-11, for this transport of ALG-1 to the ER. However, when you are in conditions where RAB-6 is maintained in an active state, it would excessively shuttle ALG-1 to the rough ER. And in this case, there would be some unfunctional, unloaded Argonaute that would accumulate and can't repress properly its targets. So with that, I'll thank the people from the lab and everyone who helped with this project, and I'll be happy to take your questions.
Thank you, Pascale. We already have one question in the chat. If you want to ask a question directly, you can click at the bottom of your screen on participants, and you will have a new window popping up on the right where you can virtually raise your hand, and I will unmute you for you to ask your question. Additionally, you can just ask question in the chat, either here or on our virtual worm Slack channel. So we can start with the first question from Jose Luis, asking if these allele were backcrossed and how many times, and if they were under the same wild type background.
Okay, so the OK allele we received from CGC, it was outcrossed 4 times before receiving it. So we just went with that one directly. The QBC allele, so it was initially obtained from the screen. So it was a mutant that had been like mutagenized, so it had many mutations. So we did outcross that 4 times. And then this was done initially in a background where we had this reporter system with the box B lambda N. So then it was crossed again to remove that reporter system. So we would just have this QBC24 mutation in an N2 background like the OK allele was.
All right, then next question from Joshua Aribe. The amount of ribosome in polysome was relatively low, with most ribosome being monosomal. Any idea why polysome were collapsed in your sample? Do you think it's a technical thing or a feature of the strain background or something else?
Yeah, okay. Well, if you compare to wild type, the total level of translation is not affected. And so this is, I think this is just a technical issue because this isn't this old machine that do these profile with a crayon on paper. So as you can see, like here, the ribosomal subunits, they were, you actually can't see them because it was too saturated. And so it was just going off the paper. So I think this is why the profile maybe looks a bit weird, but we have wild type in comparison and we don't see much difference between the two profiles.
Okay, but just a quick question for someone which has done more bacteriology than worms. What's a polysome?
Okay, so the polysome is when you have many ribosomes on the same target. So when something is actively transcribed. So the more you go into heavy, the more ribosomes that are accumulating at the same place.
All right, I see.
So if that's clear, yeah.
Yeah, thanks. And then another question from John Paul Ouyang. Did the RNAi of other RABs suppress the TBC-11 phenotypes?
We did not test other RABs. It would be interesting to look. We kind of stopped when we saw that RAB-6 had an effect, and it kind of made sense with what was known in the literature. But yes, it would be interesting to maybe test the other RABs, see if there's many that are contributing to this phenotype.